Disulfide isomerase is an enzyme that catalyzes the formation and breakage of disulfide bonds. Found in ER. RES is a small GT-PASA protein cycling between an inactive GDP-bound state and an active one, anchored to the plasma membrane through a lipid modification, prenylation. RTKs phosphorylate tyrosine residues on intracellular proteins. When a signaling molecule binds to an RTK, it induces dimerization of the receptor, which activates its intrinsic tyrosine kinase activity. This leads to phosphorylation of tyrosine residues on the receptor itself and on other proteins, initiating a signaling cascade. Small, hydrophobic signaling molecules, such as steroid hormones and thyroid hormones, can directly bind to intracellular receptors. Activation of cAMP-dependent protein kinase. Hormone binding, G-protein activation, adenylyl cyclase activation, protein kinase A activation, phosphorylation of target proteins. Ubiquitin tags proteins for degradation by the proteasome. Insulin synthesis and processing, transcription and translation, signal peptide cleavage, disulfide bond formation, Golgi apparatus processing, storage in secretory vesicles, exocytosis. Epinephrine binds to a receptor on liver cells, activating a signaling pathway that breaks down glycogen into glucose, increasing sugar levels. Allosteric inhibition. A molecule binds to a protein at a site other than the active site, changing the protein's shape. Binding of ADP to phosphofructokinase. 1. In lac operon, the repressor protein binds to the operator site, preventing transcription. Lactose binding to the repressor inactivates it, allowing. YEF2 delivers aminoacyl tRNAs to the ribosome. GTP hydrolysis by YEF2 is essential for this process. Low GTP levels could limit the rate of protein synthesis. RAS is an oncogene because mutations in RAS can lead to uncontrolled cell growth and cancer. Glucagon stimulates glycogen breakdown in the liver, releasing glucose into the bloodstream to increase blood sugar levels. Reception. A signaling molecule binds to a receptor protein on the cell surface. Transduction. The signal is amplified and transmitted through a series of molecules within the cell. Cellular response. The signal triggers a specific cellular response, such as gene expression or enzyme activation. Paracrine signaling. Signaling molecules act on nearby cells. Endocrine signaling. Signaling molecules are released into the bloodstream and act on distant cells. Autocrine signaling. Cells respond to signaling molecules that they themselves secrete. A ligand is a molecule that binds to a receptor protein, initiating a cellular response. Aldosterone is a steroid hormone produced by the adrenal cortex. It increases sodium reabsorption and potassium secretion in the kidney, helping to regulate blood pressure. Nitric oxide, NO, is a gas that diffuses into smooth muscle cells, activating guanylyl cyclase, which produces cyclic GMP, cGMP. cGMP relaxes smooth muscle cells, leading to vasodilation.